And I think it'd be good, like we were talking about, we'll do a little bit of review of what we've been seeing so far. We'll do that with a bit of the crash course. Let's bring that one up.
Our time lately talking about eating and digesting and metabolizing food. And those are some of my favorite things in the world. It's been a really great time. But as with all good parties or brunch buffets, in the end, we're left with a mess. And I'm not talking about spilled beer and Dorito crumbs. I'm talking about toxic levels of garbage that need to be cleaned up before they kill you. In your body, a lot of the cleanup that comes after metabolism is handled by the liver, which plays a tremendous role in directing dead cells and leftover chemicals to the digestive and urinary systems. But your liver can't actually escort waste out of your person. Your lungs can lend a hand exhaling carbon dioxide, and of course your colon will eventually poop out unusable stuff and old cell parts. But much of your chemical waste still needs to be sorted and disposed of, so one system steps in to bat cleanup. And that is your urinary system. This system, and specifically your kidneys, does all sorts of important homeostatic stuff like regulating your water volume, ion salt concentrations, and pH levels, and influencing your red blood cell production and blood pressure. But its main purpose, what we're going to be focusing on for the next 2 lessons, is how it filters toxic leftovers from your blood, like nitrogenous waste made by metabolizing protein, and ferries it out of the body. And spoiler alert, this all involves the how and the why and the what of your pee. Now, you probably know that kidneys are filters, and you may imagine them as sieves that strain out the bad stuff, leaving it sitting like a hairball at the bottom of the bathtub. But that is, in fact, kind of the opposite of what you should be thinking. Most of what's in your blood is totally removed by... Instead, each one is made up of about 1 million twisty microscopic filtering units called nephrons. Structurally and functionally, nephrons are where the real business of blood processing, which like pee making, begins. In 3 steps: filtration, reabsorption, and secretion. Each nephron consists of a round renal corpuscle that resides up in the cortex, followed by a long and winding renal tubule that loops around between the cortex and the medulla. The outer part of the corpuscle is a cup-shaped feature called the glomerular capsule, because inside it there's a whole tangle of capillaries called the glomerulus. That's from the Latin word for ball of yarn, which is pretty much what it looks like. And the endothelium of these capillaries is very porous, so they allow lots of fluid, waste products, ions, glucose, and amino acids to pass from the blood into the capsule. But they block out bigger molecules like blood cells and proteins, so they stay in the blood and exit through the peritubular capillaries, also known as the vasa recta. Now, all the stuff that gets squeezed out of the blood into the glomerulus is called filtrate, which is then sent along the elaborately twisting 3-centimeter long renal tubule. Even though it looks like just a tube, it actually has 3 major parts, some of which are permeable to certain substances but not others. First is the proximal convoluted tubule, or PCT, which is about as convoluted looking as its name suggests. Then the tube drops into a dramatic hairpin turn called the nephron loop, or the loop of Henle, a term that I like better personally. And finally, it ends in the distal convoluted tubule, or DCT, which empties into a collecting duct. All this twisting might make the tubule look like super inefficient, but it actually serves an important purpose, as you might expect. Just like with your small intestines, the long, curly shape of the nephron provides more time and space for it to reabsorb whatever usable stuff it can. And this meandering path also allows the parts of the tubule that are toward the end to have an effect on processes that take place closer to the beginning as they pass each other. Because a lot of the stuff that winds up in the tube are valuable commodities, like ions and glucose and water, and we don't want to just pee all of them out if we can help it. So let's trace the whole process, starting at the top with the proximal convoluted tubule, or PCT. The walls here are made of cuboidal epithelial cells with big ol' mitochondria that make ATP to power pumps that pull lots of sodium ions from the filtrate using active transport. These cells also are covered in microvilli that increase their surface area and help reabsorb much of the good stuff from the filtrate back into the blood. The remaining filtrate passes from the PCT into the loop of Henle, which starts in the cortex, then dips into the medulla before coming back into the cortex. And the form of this loop is key to its function, because its primary task is to drive the reabsorption of water by creating a salt concentration gradient in the tissue of the medulla. It does this mainly by actively pumping out salts in the ascending limb. This creates some very salty interstitial fluid in the medulla, so when new filtrate comes down the descending loop in front of it, water passively flows out and into the super salty interstitial space. Since most of this water is picked up by the blood pretty quickly, the saltiness of the interstitial space doesn't get diluted, so it can keep drawing water out of the next batch of filtrate in the descending limb. Needless to say, this is super important, because if we peed out all the water that went into our kidneys, we would die of dehydration really quick. But even after all that, we are still only 2/3 of the way through the process. As we move out of the loop of Henle into the distal convoluted tubule and onto the collecting duct, the remaining filtrate is now officially urine. But there's one more component that we have to squeeze the most out of before we excrete the stuff. Urea. Even though we think of urea as a waste product, just one more part of that protein shake that has to be dumped, the kidneys actually need it. They use it to ramp up the concentration gradient earlier in the process, making the medulla even saltier for the filtrate that's back there going through the ascending limbs. When the final steps, after the filtrate leaves the DCT, it enters the collecting duct, which runs back into the medulla. And while the salt passively draws even more water out of the collecting duct, some urea passively leaves the urine as well, making the medulla even more salty, and in turn more effective at drawing out water from the ascending limb a few steps back. So there's essentially a traveling pool of urea that escapes the urine, finds its way back into the loop of Henle, and then runs the whole course again back to the collecting duct, an ammonia-scented cycle called urea recycling. Now all that's left is a kind of last call to selectively sneak out any extra waste like hydrogen, potassium, and certain organic acids and bases using active transport. This is called tubular secretion, and it transports only select kinds of waste that have already made their way into the blood that's in the paratubular capillaries ready to leave the kidneys. This stuff is kind of like emptying your pockets of any last wads of tissue or crumpled receipts as you're walking a bag of trash to the curb. And that's how your kidneys clean up the mess left over from the giant party that is you metabolizing food. So if you thought that your kidneys were just a kind of fine mesh that filtered out bad stuff, now you know that that's not true. If you thought your urinary system was basically a matter of water goes in, pee goes out, that's definitely not true. And if you thought we were done talking about your urine, that is also not true, because next time we're going to be learning how your body regulates what's absorbed and what's excreted, and we'll find out what can happen when that regulation goes awry. But for now, you learned the anatomy of your urinary system and how your kidneys filter metabolic waste and balance salt and water concentrations in the blood. Specifically, you learned how nephrons use glomerular filtration, tubular reabsorption, and tubular secretion to reabsorb water and nutrients back into the blood and make urine with the leftovers. Thank you to our headmaster of learning, Linnea Boyev, and thank you to all of our Patreon.
Okay, so that gave us a little bit of a review of what we were talking about in terms of our nephrons. So we can give ourselves a step-by-step process in terms of the different parts of the nephron, and then kind of reviewing kind of what's going on during each part. Before I do that, drawing out on the board, did you have questions about what you were seeing or hearing in the crash course video? Dead silence. Is it no? Yeah.
How often does it recycle it, or it cycles it twice or once through the whole cycle?
The urea recycling, is that what we're talking about?
Yeah.
So that is kind of a continuous process that's occurring between the collecting tubule and then over to the tissue surrounding the loop of Henle. So it's kind of continually doing that. But while it is cycling some of it to create that concentration gradient, to help with that gradient, we are still getting a lot of that urea out of the system as well. So much like when we're talking about the filtering that's going on in the glomerulus, right? Not all of the fluid from the plasma is dropping through the filter, right? Because otherwise then there'd be only cells and, dry things continuing on in those efferent arterioles. So it's only a small portion of it that is being filtered. It's only a small portion of that urea that's being reabsorbed and then kind of recycled that way. A decent percentage of it, like 50% of it, is going into the urine so we can excrete it from the body and get rid of it. Okay, so let's take a look then in terms of our nephron and just go through a little bit of the anatomy of it as a reminder. So we've got a tube going in, an inflow pipe and an outflow pipe, and then we've got this big ball of capillaries here like that, right? So what's this inflow pipe called? Right? So that's our afferent arteriole that we have right there. So that's an arteriole. And as we were talking about last time in terms of regulating glomerular filtration, because this has smooth muscle in its wall, we can constrict it, we can dilate it, which then affects how much blood is arriving in the glomerulus and how much filtering is going to go on there. So the filtrate is going to kind of exit, and the rest of the blood is going to go out the drain, if you will, or the outflow. That's going to be our efferent arteriole. Again, in this case, we also have smooth muscle in its wall, and so we can control its diameter too. So we can control the inflow and the outflow, which is going to affect our glomerular filtration pressure and the amount of filtrate that's being produced there, right? So just like if you're in the shower, you can control how much water pressure is coming in by your dials, by your controls and stuff like that. And then with the drain, to a certain extent, you can control the outflow as well by cleaning the drain, getting the hair and muck out of there and opening it up, or letting that accumulate, and then there's going to be a backup, right? But in our body, it's really important that the afferent arteriole is bigger than the efferent arteriole so that we have that increased pressure here, that backup in the glomerulus, which creates that extra pressure to push water and small solutes through that filtration membrane, right? And that filtrate is going to be collected in the surrounding. Oh, that's a bad one. I don't like that. But maybe that's going to be my option. Otherwise, I go orange. I could go orange. Okay. Because that's just like gray, and that's not particularly good. So surrounding that is going to be a capsule. And remember, this is going to be called our glomerular capsule. Not the renal capsule. That's the big one, connective tissue on the outside. This is going to be our glomerular capsule here. So the glomerular capsule is going to be collecting the filtrate that is pushed through the filtration membrane, leaves the blood, collects in that coffee pot or that cup that surrounds it, right? Then from there, we get to this windy, windy, windy section, right? What is that section called? So it's definitely a convoluted tubule because it goes back and forth and back and forth. And it's a small tube, so we call it tubule, but it's close to the glomerulus. So we call that a proximal convoluted tubule, or PCT. And then from there, we get to this really weird thing that goes down and goes back up like that. What's that called? So you can call it loop of Henle, or you can call it the nephron loop. Doesn't matter. Each one is fine. And then from there, it gets windy, 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 windy again. Now that's going to be our distal convoluted tubule because it's further out in terms of the flow. Now realize that I'm spreading this nephron out because it's easier to draw that way. But we've seen in the models and in the microscopes, in fact, this proximal or this distal convoluted tubule comes right back by the beginning of this nephron and helps to form our juxtaglomerular apparatus, right? Our checkpoint. Because this is kind of the beginning, and this is almost the end. That's the end of our nephron. And so it's good to be able to check and see how this nephron is doing in terms of processing our fluids and our solutes. And this is going to dump into a very large diameter tube, which is going to be our what? Collecting tubule. And it is going to be collecting a whole bunch of filtrate or urine from a whole bunch of different nephrons, right? So it's technically not part of one nephron because it collects from a whole bunch of other ones. Okay, so now we've got our monster nephron there. And we know that in terms of our processes, for our processes, we've got our filtration. We've got, what's the second one? Reabsorption, where we're getting it back from the tubule and putting it into the capillary, and then we have our secretion, where we're pushing it from the capillary into the tubule. And so filtration, that one is going to be occurring here. It's often referred to as glomerular filtration, so that's the only place where it occurs. Reabsorption occurs here, mostly, because, right, that's the first place where we're going to need to get things back. It also occurs here. It also occurs here. And it also occurs there, right? So we get filtration here. We get reabsorption here, reabsorption here, reabsorption here, reabsorption here, okay? Now, I'm broadly saying reabsorption, but we know that there are different things that are reabsorbed. Water is an important thing that's reabsorbed via osmosis, and then sodium, and then there's other things, even like urea and stuff like that, right? So we have different examples of things that are reabsorbed that we do need to know. Then we have secretion, Not here. Yep. We get it here. Yeah. We get it even a bit here, a little bit, as well as we transition into our collecting tubule and our distal convoluted tubule. We don't get it here, really. So a lot of reabsorption going on there. All right, so we've kind of mapped out our functions now on our nephron. So now what we were talking about last time is the purpose of this nephron loop. Because we had an idea with the proximal convoluted tubule, this is a main place for reabsorption and secretion. Because we said that the formation of filtrate at the beginning is just a size selective process, right? Small things are whoop, dropping through the holes. And a lot of those small things are very important that we need to get back, so we're going to reabsorb them right away in the proximal convoluted tubule, which is why we have all those microvilli lining the cells of the proximal convoluted tubule, giving us more surface area. A lot of secretion there too. First place we're going to push extra things out. Potassium ions, hydrogen ions, stuff like that. Push it into the filtrate to get rid of it as well. This is kind of cleanup crew. If we need to do a little bit more, we have some options of doing that before it's totally out of the nephron. And then this is where we were talking about some unique things going on in terms of why we have the shape of the nephron loop and what it's doing. So with that nephron loop, we want to go left, we want to go right, who knows? With the nephron loop itself, we'll go over here. It's got two parts to it. It's got a descending limb where the fluid is going down from the proximal convoluted tubule, and then it's got an ascending limb where it is coming up, leading to the distal convoluted tubule. Not lib, but limb, like that. Now, in the ascending limb, we're doing some reabsorption. We said reabsorption occurs in that tubule. In the ascending limb, what we are reabsorbing also has an A in it. We're reabsorbing mainly sodium. Technically, there's some chloride following it and stuff like that, but we're focusing on sodium there. So the reabsorption that is occurring here is we are using little pumps to reabsorb sodium. Boink. So that's the main role of the ascending limb of the loop of Henle. We're reabsorbing sodium. Okay? And as we're doing that, it's going to make the surrounding tissue here more concentrated because we're piling solutes into it, right? So it's making the concentration in the surrounding tissue of the kidney here more concentrated. And the reason that's important is that on the descending limb, we are going to be reabsorbing water, which has an E in it. So it's descending. And so on the descending limb, where it's coming down, all of that sodium that we are reabsorbing and piling up out here in the surrounding tissue is creating an osmotic gradient. The water that's coming down the descending tubule is looking around and go, oh, there's a whole bunch of sodium out there and not a lot of me, so I'm going to diffuse out, right? Through osmosis, I'm going to try to balance things out. And so on the descending limb, we've got water that is being reabsorbed. And the reason that the water is coming out is because of the sodium that we're pumping out on the ascending limb, right? So these two parts of the loop of Henle, even though I'm drawing them out on the board and they're like this far apart, in the microscope you'll see they're right next to one another. So what we are doing in that ascending limb, reabsorbing sodium, is creating an osmotic gradient to draw off the water like that, to allow us to reabsorb water out of the descending limb. And that's some of our, what's called, right, we had two types of water reabsorption. We had obligatory water reabsorption, we have to get it back. And then we had facultative, optional water reabsorption, where like later on, as we get towards the collecting tubule, it's like, do we need more water? Okay, we do, we'll pull some more. If we don't, let it go, right? Optional water reabsorption later on. This is obligatory water reabsorption happening all the time to make sure that we're not a dried out husk, right? So what we're seeing here is that the two parts, the two limbs of the nephron loop are a little bit different. Sodium being reabsorbed on the ascending part, water being reabsorbed on the descending part because of that sodium that we saw there. So that is what is going to create a gradient, a concentration gradient in the medulla, because we're dipping down to the medulla. It's going to create a concentration gradient in the medulla, because what happens here is, remember like on the model, we have the tubule like that, and then we have all these paratubular capillaries, right, surrounding all of this. And surrounding the nephron loop, we've got the vasa recta. We have capillaries there. And so the blood that's coming down and wrapping these tubules here, those capillaries are picking up a lot of that water. When we say we're reabsorbing it, that water actually is picked up by the blood and is carried away. So the concentration gradient is still out here. It's still a very concentrated environment because the water that we're reabsorbing, we're whisking it away by the vasa recta vessels that are surrounding it. So what we're seeing here is this right here. This loop of Henle is creating a gradient of anywhere from 300 milliosmoles at the top down to about 1200 milliosmoles down at the bottom, down in the deepest part of the medulla. So it's creating this gradient as we reabsorb this sodium and pile it up in the rest of the kidney tissue like that. So this nephron itself creates the gradient, what's called the vertical osmotic gradient. So it creates they call it vertical because it goes from between the cortex and the medulla, osmotic gradient. Okay? So that's the role of the nephron loop. It creates a concentration where it becomes more and more concentrated as we get down here, down into the medulla. And the reason that's important is that after this fluid, this filtrate continues on and arrives here at the collecting tubule, we said here in the collecting tubule is where we have our facultative, our optional water reabsorption. Now, the only way that we can reabsorb water is if we have an osmotic gradient, if it's more concentrated on one side of the membrane compared to the other. And this fluid, this filtrate, as it's coming up here, right, as it comes up here, it comes to about 300 milliosmoles, about the same concentration as it started. It can be even less, even down to like less than 200 milliosmoles. So this is very dilute filtrate or urine as we're entering into the collecting tubule. And in the collecting tubule then, this is where we need to look and decide, okay, do we need to get rid of this excess water if I've been drinking a lot? I need to get rid of water. And if that's the case, we're just going to let that 300 milliosmol water, that dilute water, watery filtrate, go out and exit the body. You're going to be producing that urine that looks just like water, right? But if you are dehydrated, this is where we can reclaim even more water under the influence of a hormone that's called antidiuretic hormone. So if we are dehydrated, your pituitary gland is going to get that information, right? Blood pressure is getting low maybe and things like that. The blood's getting really concentrated. It's going to release antidiuretic hormone, right? Antidiuretic hormone basically means no pee hormone, right? A diuretic makes you pee. Antidiuretic, we're not going to pee as much, right? So antidiuretic hormone, when this gets released, when you're dehydrated, what's going to happen is that in the collecting tubule, we're going to insert these little channels here that are called aquaporins, right? It has it right in the name that these are going to be little pores for water. And so now that we've inserted these little channels, these little pores for water, the water that's coming down here sees this concentration gradient, right? So it's 300 milliosmoles. As we get down here, it's like 400 out here, 500, 600. It's really concentrated out here, and the water will leave and diffuse out via osmosis because it now has those little channels, those little aquaporins like that, right? So the collecting tubule, under the right conditions when you're dehydrated, will use the gradient, the concentration gradient that the nephron loop has created to be able to reabsorb even more water than we reabsorbed here, even more water than we reabsorbed there. This is obligatory water reabsorption. This is facultative, optional water reabsorption. But the only reason that we're able to reabsorb extra water down here is because the nephron loop has created this concentration gradient from the cortex down into the medulla, and now the collecting tubule is going to use that gradient. right? So that's the whole reason that the nephron loop is doing what it's doing. The nephron loop creates that concentration gradient that the collecting tubule can use to reabsorb water. Okay? How is that? Better? Okay. So the key parts about that, because there is an exam objective that talks about, oh, how do we produce dilute urine or concentrated urine, and what are the structures involved? You would need to include these two things in it, right? You would need to include the nephron loop, explaining that the nephron loop creates this concentration gradient from the cortex to the medulla through its reabsorption of sodium on the ascending limb, driving the reabsorption of water on the descending limb. And then you would need to also explain that the collecting tubule here uses that gradient as long as there's antidiuretic hormone providing stimulation to create these little aquaporins so that we can reabsorb more water as necessary. Yeah?
So the gradient, is it kind of like a dilution?
It's more of a concentration, right? Because it's more diluted, it's less concentrated up here, and it's more concentrated down here. So in that way, as we go down here, because that way, this filtrate that is entering the tubule, and it's going from the cortex to the medulla, as it migrates down, right? These are within those pyramids that we saw in the kidney. As it's coming down there, it's entering an environment that's more and more concentrated. So that's more and more of a draw to reabsorb that water, if we can. Now, the only way we can reabsorb that water is if these aquaporins, these channels are inserted there. If we're not dehydrated, that means no ADH, no channels, no aquaporins. That means water's shooting down there and it's looking there and it sees that concentration gradient, but it can't get to it. There's no channels to allow it to be able to reabsorb. So the water stays in the tubule and ends up being peed out instead. So that way, that's the way that your kidney can tweak or modify the urine concentration. Okay? So it has this kind of mechanism that's under the control of hormones, antidiuretic hormone, which allow it to reabsorb extra water here in the collecting tubule under what's called our facultative water reabsorption, because that nephron loop is creating that gradient to do so. This is like A Beautiful Mind or something like that. You guys seen that movie? It's an old movie now. Okay. So what we're seeing here is a much nicer picture and depiction of what I was talking about, right? So you can see some of those numbers that I was talking about and how there is the reabsorption of water on the descending limb like that as we go down into the medulla. And that is driven by the reabsorption of sodium that we see here in the ascending limb. Okay? Okay. So as we mentioned, if we're going to have dilute urine is where, we don't need this antidiuretic hormone. We don't need these aquaporins. And so this very dilute urine that enters in the collecting tubule, the concentration stays the same. It just shoots out, no extra reabsorption of water occurring there, right? But if we are needing to form concentrated urine, then that antidiuretic hormone, I abbreviated it, but antidiuretic hormone, is going to cause the insertion of those aquaporins. I should have written that on there. This is an aquaporin. It's probably in there somewhere. And in that case, we get to reabsorb even more water there. That is going to retain fluid and then make sure that our blood volume stays up, make sure our blood pressure stays up, right? Because we're in this dehydrated state. We don't want to allow the loss of any extra fluid. So that is our facultative, optional water reabsorption under the control of antidiuretic hormone. And then the kidney responds by reabsorbing that extra water. Okay? Okay. So, just wrapping up on that, because this is the last part of the nephron, which is doing all of that work, like we said. just wrapping up with that, as we were talking about, right, a diuretic makes you pee. So diuresis is where you are peeing too much, you're losing water instead, and there are a variety of chemicals that can affect this process. So for instance, I'll go jump to the ones that you're most concerned about, alcohol and caffeine, right? So alcohol and caffeine, especially, right, alcohol inhibits the release of antidiuretic hormone. Oh, so no antidiuretic hormone means that no extra water reabsorption is going to occur here. That means we're going to be losing a bunch of extra water, right? Even though we're drinking, right? I don't know why I'm dehydrated. I drank all night, right? But you're drinking something that is a diuretic. It's preventing your nephron from reabsorbing that extra water that it needs to. So you're losing more fluid than you're gaining. So that ends up being a deficit, right? Caffeine, as well as other diuretics, inhibit sodium ion reabsorption. And we know that sodium ion reabsorption here is what's creating that gradient. So if that sodium reabsorption is occurring, that means less water reabsorbed here. That means the concentration gradient isn't as strong. That means less water reabsorption there too. That's a double whammy, okay? So if you are drinking some sort of Red Bull and vodka, that's a double whammy. Those are both diuretics, right? So you're going to be severely dehydrated and you're going to be awake to enjoy it, right? But in terms of other diuretics, we know that this helps to regulate blood volume and blood concentration, right? By making sure that we can retain or excrete water as necessary. But if you have a problem with hypertension, your blood pressure is too high and it's sustained that way, one of the ways to treat, not the cause of it, but the symptom, is to get rid of some of that excess fluid. So if you are peeing out a little bit more, that means your blood volume goes down, your blood pressure goes down, and now you've kind of solved the issue. You haven't, again, solved the cause of it. You don't know what the cause is necessarily, but at least your blood pressure is down and your blood vessels are not so stressed. So a lot of the diuretics that they may give you, sometimes they actually call them loop diuretics. This is the loop that they're referring to where they affect the sodium that's being reabsorbed there. Yeah?
So with alcohol though,
Yeah.
when they're trying to like rehydrate you, don't they use also like potassium in those like solutions?
Yeah, so it's certainly possible when you're talking about adding fluids back to somebody, right? Some of the things that we're talking about reabsorbing and secreting, we want to make sure that things are always in balance. And so when you're adding things back to somebody as well, you got to think, okay, what have they lost? Certainly, they've been losing water, but we also tend to lose some of these electrolytes, these solutes as well. And most notably, as we've been talking about, I really wish I had like a green one. I don't know if somebody really likes the long wavelengths.
That has a blue one.
Yeah.
Can you catch?
Yeah, we'll find out.
All right.
So now I'm so distracted by this really cool one here. I don't even know what we were talking about. But the sodium that we're reabsorbing, right, is often due to, the mechanism is a sodium-potassium pump, right? So sodium-potassium ATPase or whatever, right? And every 3 sodium that we are kicking out this way, that we are basically reabsorbing, like we're seeing over there, we are losing or secreting 2 potassium, just like we learned in action potentials and stuff like that. So the sodium reabsorption that's going on, that's good, okay, we're getting that, but then we are losing potassium at the same time. So sometimes when we're rehydrating somebody, not only do we want to look at the concentration of what we're putting in and make sure that it is isoosmotic, right? But we want to make sure that if there are certain electrolytes they may be losing, that we're replacing those too. And that's why they say if you're exercising a lot and doing things, you got to think the sweat and the urine that you are losing, it's not pure water. There's always going to be salts with it because the salts, even, right, and any electrolyte is considered a salt. There's potassium chloride, there's sodium chloride, stuff like that. The salts are drawing the water, and so you got to think about replacing those electrolytes, those salts as well. Yeah.
So is that, or why do people, when they're so hydrated, if you try and take their blood pressure, like their whole hand squeezes up and tightens? Do you know like why that happens? Electrolytes loss? I don't know.
I don't know.
I
So
see it.
when you're taking their blood pressure, yeah?
Yeah, if some people come in because they've been throwing up or
Yeah.
severely dehydrated, it'll contract their wrist first, their hand first, and then their wrist and sometimes their whole arm if they're dehydrated enough.
I would have to think about that. So we know that the idea here is that, like with the digestive system, et cetera, that's where we're gaining our fluids, and we're going to be talking about that in the next chapter. We're gaining our fluids there. So in this particular case, we've been losing fluids. We haven't been gaining a lot of fluids. And then the fluids that we gain go into the bloodstream. Bloodstream delivers it to the tissues, like the muscles. So if you're dehydrated, not only is your blood volume and blood pressure low, but the tissues that are normally receiving that fluid are also strained, right? They haven't been receiving the fluid. So it could be that that muscle tissue is also dehydrated. And I don't know, this is total, conjecture, but like when we're doing blood pressure, you're constricting new blood getting to those muscles, right? So you're blocking off the brachial artery, et cetera. And so it's possible that now all of a sudden they're a little bit more even dehydrated more because we're restricting any more fluid getting to them. So they're already in this stress state. And so for a period of time, maybe they're like, it's kind of like letting the air out of the balloon. You're kind of not giving them things that keep them nice and plump, and they're already kind of small, and now they're getting smaller. And so maybe as they get smaller, they shrink and contract a bit. I don't know.
They're like, maybe they're already so stressed out or so low on those resources that we're constricting, like you said, the blood flow and the resources.
Yeah. So they just might be temporarily shrinking.
Just curious.
Yeah, interesting. Yeah.
High glucose levels.
Oh, yeah.
And also, is that the reason that diabetics as symptom can be thirst?
Yes, exactly. So we'll talk about this when we get to endocrine system, right? Regulating glucose, we talked about this in urinary system as well. So normally, glucose is small enough it slips through the filter. So we have these transporters, sodium glucose transporters, where we pull it back. And normally we can get 100% of the glucose that is filtered out, we can reabsorb it. Unless, like we said, there's something that's at an imbalance. Like this past weekend where we were ingesting lots of glucose. Now the glucose levels in the blood are so high, there's so much ending up in the urine, we can't reclaim it all. Or if there's somebody who doesn't have enough insulin or their cells aren't responsive to insulin, their blood glucose levels are high as well. So there's a lot of glucose showing up in that filtrate. Now, much like we were talking about the concentration of the filtrate here and how concentrations of solutes can draw water, if there's more glucose in this filtrate, then this is already more concentrated. More water is going to tend to stay with it rather than being reabsorbed via these other mechanisms here. So if this is like 500 milliosmoles because there's a bunch of extra sugar in that because you're diabetic or you've eaten a lot of Easter candy, then there's less of a gradient here between the concentration inside the tubule and outside. So more of it's going to stay in and less of it's going to be able to be reabsorbed, right? Because when we have a diffusion gradient, the steeper the gradient, like a ball rolling downhill, the faster the diffusion. So if we have less of a gradient, because there's a bunch of sugar in the filtrate, in the urine too, now the water is going to tend to stay there and not diffuse out. Yeah. Okay. So, yeah.
Can you just, I think I have them backwards, where obligatory and facultative occur.
Yeah, obligatory and facultative water reabsorption, two types of water reabsorption that has to occur in the nephron, right? Obligatory, we're obliged, we have to get water back so that we're not a dried up husk. That is occurring here and here at the beginning. So we get water reabsorption here, and in fact, that's why I just brought up this slide. This slide does a good job of showing you the different parts of the nephron and what's happening in each part. So what we can see here, this is the proximal convoluted tubule. See how many things are being reabsorbed. If the arrow is going from the tubule out, that's reabsorption. And if we're seeing arrows going into the tubule, that's secretion. So we get a lot of reabsorption of water right at the beginning. That's obligatory water reabsorption. We get obligatory water reabsorption coming down the descending limb of the loop of Henle. We see it right there. And then the optional water reabsorption tends to occur in our last tubular segments here, in the collecting tubule, And then it kind of depends on the textbook as to whether it occurs a bit in the distal convoluted tubule too. They think in some cases you might have aquaporins and stuff like that that can happen in a distal convoluted tube. So the obligatory water reabsorption happens right away, right after we filter it out. We get it back in these first tubular segments. The facultative optional water reabsorption occurs in the last tubular segments, where it's like, okay, what do we want to do with this water? We're leaving the system. Do you want it? Do you want it? Do you want it? Okay, we're letting it go. Or if we have antidiuretic hormone, yep, we're going to pull that back as well. Mm-hmm. Okay. So that at least gets us out of the kidney. Woo! I told you, kidneys are crazy. You look at them from the beginning and you're like, that's easy. It's just like a jelly bean. It's jelly bean with a straw coming out of it. That's good. And all it does is filter blood and it just drops right out like a coffee pot, right? But then we learn that these three processes are going on in different segments, and each little segment has a different role to play, right? Kind of like digestive system, right? Different stops along the way have a different role to play in terms of the function, okay? But the easy part now, as we'll finish up urinary system, is just the rest of it. Ureters, bladder, urethra, okay? So ureters, as we mentioned, are tubes that are going to be draining the kidney and taking it down to the bladder. So we can see them along the sides there. They enter the posterior side of the bladder, as we've been seeing in the models, along here and along here. And interestingly enough, so these are also retroperitoneal. There's that term that I was talking about, behind the peritoneum. And they go into the posterior side of the bladder. And because of the angle that they come in, you can see them coming in almost horizontal like that. They kind of almost have like a physiological valve like that. It's not like a sphincter like we've seen with the food tubes, but it is a bit of a valve where we want to make sure we're having this flow go from the kidney to the ureter to the bladder and not back up, right? So we get the flow coming down here, and it's not just gravity. It's not just that the bladder is down in the pelvis. It is that these are muscular tubes that we'll see in the microscope. So we have circular muscle and longitudinal muscle in there, and they are doing peristaltic waves, right? They're squeezing it down from the kidney, from the renal pelvis down to the bladder to make sure it exits there. So you can even get the urine being produced and transferred to the bladder if you were hanging upside down, which is important, right? Because we don't want it backing up to the kidneys itself, because there's always more that's going to be showing up. So just like we have more blood showing up back to the heart all the time, we have those valves that make sure that it's a one-way flow through there too. Okay, so these ureters come to the bladder, and now we dump into this big storage organ, much like the stomach. And so like the stomach, we can see that there are rugae in it, right? These wrinkles like that. And where those ureters dump in, that's the entry point. There are two of those, and then there's an exit point down here. So it makes kind of a triangle that we learned is called the trigon inside of the ureter. And here as well, it's not just a passive process, right? We know that there are sphincters, again, like the end of the digestive system. There's one that is involuntary, an internal one that is smooth muscle. And then there's one that is a voluntary one that's extrinsic, made up of skeletal muscle, voluntary, so you can control when you drain the bladder. But that drainage is not just passive. It's not just like there's a straw there again. We have a muscular tube, and we have a muscular organ. That bladder has a very thick wall to it called a detrusor muscle. And so when you're emptying the bladder, you're actually squeezing the bladder to push that fluid out. And that's called micturition or voiding, okay? So these muscles are going to make sure that on the outside that we're releasing urine as necessary, but there's also muscles throughout, muscles in the ureters, muscles within the bladder that are going to continue to move the urine as far as it needs to go, and then we'll have our voluntary control of that when we're emptying the bladder itself. So when we are doing this voiding, what we're going to do is we're going to stimulate the detrusor muscle in the bladder. We're going to start squeezing it to push it out, and then we're going to inhibit those sphincters. We're going to loosen the sphincters, the bands on the urethra, to allow that fluid to exit as well. So like anything else, like we talked about with the stomach, right, there are going to be stretch receptors in there that tell us about how full it is. You start getting that sensation of, oh, I'm going to have to go pee soon. No, I can put it off, right? You still have voluntary control up to a point, right? At some point, there's going to be so much fluid in there and more coming down from the kidneys all the time that you're not going to be able to hold it, right? Any skeletal muscle that you have fatigues after a while. So you put it off too long, then there's going to be a problem, right? And toddlers and, that are learning to control that, it's just like any other skeletal muscle. It takes practice. It takes practice for them to be able to talk and use different muscles of their tongue and their lips and things like that. It was practice for them of skeletal muscles to walk and balance, and it's practice using skeletal muscles to control that outflow, both of the digestive system as well as the urinary system, right? Yeah, back there.
This is
Coming back.
random. How does UTIs work in this? Like, would that be considered with the anti-diarrhea one?
So that's a good question as well, right? So a UTI is a urinary tract infection. Now this, as with many of our epithelial-lined tubes, opens up to the outside, right? So we're thinking about things exiting, but to be sure, at the surface here, we know there are lots of bacteria and things like that on the surface of our body that are just waiting to try to gain entry to the body. And so if there are a lot of bacteria that are entering and kind of taking up residence there, then you can get it along the urethra. It can migrate. Now you get an infection of the bladder. And now it might even migrate, right, because they're all connected, up to the ureters. And you could even get it going up to the kidneys as well. So the UTI is going to be infection of this pathway. Now we do have, right, lymphatic tissue and things like that. We know that this system is flushing itself out regularly, so there are things that can help that. But sometimes we get out of whack and our immune defenses aren't able to totally combat it. So you may need, right, antibiotics or something like that. Yeah. And in women, as we'll get to when we're doing reproductive system as well, this urethra is much shorter, right, compared to men. In men, there's actually 3 sections to the urethra as opposed to women. And the opening itself is next to the vagina, next to the anus. So all of these things are kind of co-located. So there is a much higher chance of bacteria because we know that there's lots of bacteria in the large intestine that are exiting there. And some are pathogenic, some are symbiotic. But in this, if they gain access over here, now that can throw everything off. It's just that they're so co-located, right? So they're so closely located like that. So men can also get UTIs, but it's much more common in women. Yeah, sorry.
I have a random question as well. I've heard that if you hold in your pee for too long, or if you do that very often,
Yeah.
you actually weaken the muscle, the sphincter, or something like that. Like so when you get older, you can't hold it in as well, or something like that. Is there truth to that?
That's a good question. I don't know. There's definitely, right, all of our systems change through time, right? So as we were talking about, like babies and toddlers, and then as you are, a teenager and stuff like that, all your systems change. Metabolism changes. You could eat whatever you wanted before, and now it's like, oh, well, it's low calorie on here, right? And muscles don't work the same. So same thing in terms of muscular control and weakness of muscles overall. Like you don't have generally like a 80-year-old man who's out there and, pumping iron and all racked up and stuff like that, right? Just because the body doesn't respond that way. So those muscles tend to weaken as well. And I think that's just like an age-related thing. In terms of like holding it, I don't know. I can't say for sure. You can probably go either way with that because all other skeletal muscle, the more you use it, it tends to hypertrophy and get a bit bigger. So it's a little bit stronger. And in fact, with those that have issues with incontinence, men or women, there are like pelvic floor exercises that they have you do, right? You can contract those things, and if you do that more regularly, then they may actually be a little bit more responsive. So, and everybody's different too, right? It can affect people more, so one person more, other person less, they have less issue with it.
So I unofficially heard that might be true because instead of training the sphincter, the bladder sphincter, to be able to hold together for longer periods of time, if you're going to the restroom every time you leave the house, it can detrain that sphincter so that it's not as effective, long-lasting. it
Mhm.
wants a release more often if you overuse it, is what I've heard people say.
Yeah, interesting. All right, so we got down to bladder and then urethra. Like we said, urethra, which is going to be draining it via our micturition or voiding. So the cool thing about the last part of the urinary system is that those last organs are very, very simple. They're all about just moving that fluid that we've created up in the kidney out away from the kidney, storing it temporarily until it's a convenient time to get rid of it out of the body. So there's no more filtration, reabsorption, and secretion. None of that happening in any of those other organs of the urinary system, right? At least in the humans. In other types of organisms, there are other types of organisms that can actually reabsorb water from the bladder where they've stored it. So like there's certain types of toads and things like that that'll go in, like when they go into the dry period, like in Australia, and they bury themselves in the dried up dirt. So they will take in a lot of fluid. They will hold fluid in their bladder, and they can actually get water from the bladder itself because they're not out getting fluid in this dry period. So there are other organisms that have a urinary system that's more adapted compared to ours, but ours works pretty well. Okay? Questions for urinary?
Do you have any effects that pregnancy has on the sphincter?
Yeah, so that, right, like we talked about, those organs down there of the reproductive system and the urinary system undergo stress and strain in many different ways, and age-related comes into play as well. So for the female urinary system, like we said, it's right next to the reproductive system. The vagina is just posterior to that. And so with pregnancy, what happens is, and we'll talk about this coming up in reproductive system, we're going to have a baby potentially exiting there, and a normally kind of small hole is going to need to adapt and get much larger. So that whole pelvic floor is going to have to adapt and kind of loosen and that type of thing. And generally as things loosen, like a rubber band, right? 2 times, it's like, oh, it snaps back, and then with a lot of use, it gets a little bit looser and it doesn't quite come back. So after, with age and then also after pregnancy, all those pelvic floor muscles, again, they adapt to loosen and get bigger, and then they try to come back to an original size. Even the uterus, right? Uterus is going to be about the same size as the bladder, a little bit bigger, but obviously during pregnancy, the uterus can be like this big. And after pregnancy, it tries to shrink back and does muscular contraction to try to go back to its original size, but it never totally gets there. So the same type of thing happens. It's just a lot of those muscles of the pelvic floor are then, because of their close location, are going to be affected as well and not quite performing the same. Yeah. Yeah.
So every time ADH is released or inhibited, it is actually building or destroying aquaporins every time?
Oh, so that's a good point. Yeah. So, and again, nothing that you would have to know for the lecture exam or whatever, but these channels are proteins. We know that cells manufacture proteins all the time, right, with the ribosomes and stuff like that. So they are actually manufacturing them in response to antidiuretic hormone. They've got receptors on the surface, get that signal, and then it ramps up that production. They have some inside of the cells, and what they can do is they can transfer them to the surface so that they are creating those openings. So again, we won't get into the details of it, but we'll have to have channels on this side where the water is, and on this side of the cell where it's going to be reabsorbed back into the tissue. And so we kind of keep them on this side, and then under the influence of antidiuretic hormone, we'll shuttle some that we've already made over the other side and create that opening, that full pathway for the water as well. So not only do we create more, but we kind of shuttle them to important areas of the cell that allows that passageway.
What happens once we're hydrated and no longer need those aquaporins?
So those aquaporins, they're only effective if they're at the edge of the surface of the cell. So it's like if you have a straw, that straw exists wherever it is, but it's only when you put it into the cup and it has access to the water where you can transfer water. So if we take those aquaporins and we just put them in the center of the cell, now the water can't get past that lipid bilayer anymore. So only when the channel is inserted in there, now we have that little straw-like action where it can go through. So we just draw them in as well. Yeah. Good questions. Okay, so let me look at all that in the back.